Thank you everyone for joining us today. My name is Vinh Pham, so I'm a software technical application scientist. So today I'm going to talk about metagenomic sequencing, introduction to data analysis. So this is the second part of our metagenomic sequencing series. I include the link in the resource slide for the first part, which is introduction to library prep and sequencing. So the topic that I'm going to talk about today include microbial genomics overview, introduction to metagenomics, approaches to study metagenomics. I'm going to talk about targeted sequencing and 16S ribosome RNA sequencing data analysis, and shotgun metagenomics. Especially, I will focus on DRAGEN Metagenomics App on BaseSpace Sequence Hub. And finally, I'm going to talk about briefly about the BaseSpace DRAGEN RNA pathogen detection app and also DRAGEN COVID Lineage App. Okay, so what is metagenomics? The term metagenomics first appeared in the publication in 1998 and is the study of metagenomes, genetic material recovered directly from environmental samples, and involves the sequencing of many individual microorganisms' genomes from those samples. Metagenomics has allowed researchers to create a picture of environmental microbial life without the need to isolate and culture individual microbes. And metagenomics has become an essential tool to study, diversity of microbial community, contribute to many aspects of human, animal, and environmental health. So why is metagenomics useful? This is a tool to look at the microbial communities in a deeper way. It has been estimated that more than 99% of the bacteria present in nature are non-culturable organisms. So the culture-based method has its limitations. For example, it requires live pathogen or pure culture because bacteria cannot be separated. So with the new method to study function and diversity of microorganisms, metagenomics is an expanding field within microbial ecology that provides access to the genomes of the total microbial community in any given environment. So there are two main questions in metagenomic study. The first question is, who is there? Or this answers the questions, what is the relative proportion of the community members, or what is the percentage of bacteria or archaea in the environment? And the second question is, what are they doing or how they are doing? What is exactly they are doing in this particular environment? So metagenomics is a microbial analysis tool with the best resolution. To determine the identity and abundance of microbiomes in a defined sample, there are two sequencing approaches that I want to talk about today. The first one is the targeted or amplicon-based sequencing. And the second one is shotgun metagenomics or whole genome sequencing. So with the amplicon sequencing, that relies on PCR amplification and sequencing of the marker genes. For example, 16S ribosome RNA gene to analyze the presence of archaea and bacteria, or you can use internal transcript spacer, ITS, to characterize the fungal community. The second method is a whole genome sequencing or shotgun metagenomics. This approach refers to sequencing all genomes existing in a sample to analyze the biodiversity and also the function of the microbial community instead of targeting a specific genome locus for amplification. So let's go over each approach here and focus on how we analyze the data in each method. So first, I want to talk about 16S ribosome RNA sequencing. I will mention briefly about ITS sequencing, but we'll include some resources for ITS sequencing if you are interested in fungal classification. All right, so what is 16S ribosome RNA gene sequencing? 16S ribosome RNA is a part of ribosomal RNA, which is about 1,500 nucleotide long. This is a component of the 30S small subunit. Why do we use 16S ribosome RNA? As you know, 16S ribosome RNA is using as a marker gene for classification of bacteria and archaea. The 16S ribosome RNA gene includes some highly conserved regions and hypervariable regions. So this diagram is showing those conserved regions and hypervariable regions in 16S ribosome RNA gene. Those conserved regions, which is highlighted in blue here in the diagram, help with the primer design. These regions in the orange color here is hypervariable regions. So there are 9 regions in 16S ribosome RNA gene, and they provide the specific for species classification for bacterial and archaea identification. Illumina sequencing protocol focuses on V3 and V4 variable region sequencing. So the general workflow for 16S ribosome sequencing, according to Illumina protocol, that we amplify the V3 and V4 regions using the specific primers with a length of 460 base pair. And then the product will go through library prep process using, for example, NextGenXD library prep kit. And then library pool will be sequenced on our instruments, for example, MySeq instrument, iSeq, or MiniSeq. For example, for MySeq, you can use V3 reagent kit 2x250 base pair. Next, the sequencing data will be analyzed using Illumina software that includes the 16S Metagenomics App on BaseSpace. Then that will be the one that we are going to talk about today for 16S ribosome RNA sequencing. If you are using MySeq instrument, you can also have the option to use MySeq Reporter with Metagenomics Workflow or Local Run Manager, and that will include 16S Metagenomics module. So the general workflow for 16S Metagenomics app on BaseSpace will be the pair and reads will be trimmed, the adapter or low quality reads, and then the next step will be classification step, which classifies the reads using RDP classifier that match the reads to the database. So on this app, there are 3 different databases, and we are going to talk in detail about this step in the next few slides. The output of this will be the taxonomic classification. So if you're familiar with 16S ribosome RNA analysis, there are a lot of options. 2 popular 16S ribosome RNA analysis pipeline include CHARM or MASA. However, today we are going to talk about the 16S ribosome analysis app on our BaseSpace Sequence Hub. So if you are familiar with BaseSpace, this is our cloud system that you can store your data and analyze your data with various applications. So it's not only metagenomics. For the targeted sequencing 16S ribosome, we are going to talk about this app, and in addition, we also have 16S Metagenomics database creator that you can use to create the custom database. So the 16S Metagenomics app on BaseSpace analyzes the DNA from the empty-con sequencing of the 16S ribosome RNA genes. So I include here the online help page as a reference. So if you want to read more about the instruction how to use the app and the detailed information about the classification steps. For the classification steps, this step uses the classify reads, which is the high performance implementation of the ribosomal database project, RDP classifier. And this one is using 32 basis words. So it performs the classification by counting the k-mers in each read, and then use the naive Bayes classifier to identify the best match to the database. So this is similar method in Kraken2. I include here the reference paper at the end, in the bottom of this slide. This is the original RDP classifier. So in the classify reads of 16S Metagenomics app, compared to the original RDP classifier, the classification are performed actually to the species level. The next step will be classification statistics are calculated both by sample and in aggregate, and the results are summarized in the interactive visualization. You can also get the raw classification data to download and perform your downstream analysis. as I mentioned before, for the database, the 16S Metagenomics app is using Illumina curated version of green genes. There is also RAPSIC IDP 16S v3 and also the fungal ITS database, if you are interested in ITS sequencing for fungal classification. So to launch the app, you can go to our BaseSpace and then select the 16S Metagenomics app. Once you click on Launch Application, then you can select the project, select the biosample, and here the database is available to select. And then you can note that there's an option for you to use a custom database. So you can select custom option. Once the analysis is completed, the first output that we want to take a look at the report tab. So 16S Metagenomics app provides a comparison of all samples in the aggregate summary page. This is useful when we want to perform sample-to-sample comparisons. For example, if you want to compare different soil condition or sample types or different type of treatments. The aggregate results section contains the summary files to download that will include a PDF aggregate summary report with the tables showing the same information as in the BaseSpace report. You can also find the CSV files that have the number of reads classified to each classification by sample and organized by taxonomic level. So this is example of the aggregate summary folder that contains the reads count for each taxonomic level. And then the read counts here are all raw read counts that match to each taxonomic level and not normalized. We can take a quick look at the species level aggregate count in that folder. So in these files, the columns are the samples, and each row will be the species and the number of the reads classified to that species level per sample. The aggregate summary output report also includes the clustering dendrogram. So this dendrogram shows the clustering of the sample based on the genus level classification. The bar chart beneath each other or each sample show the relative abundance of each genus level classification. So for example, if I move around this bar chart, I can see that this genus has more than 123,000 reads belong to this genus in this sample. This app also generates the summary report per sample. So that will include, the PDF summary report and also the CSV classification summary that we are going to talk in the next few slides. Also, you can see the sample information and also classification statistics are presented in the sample report. One of the interactive reports that was generated in the sample report is the sunburst chart. So the sunburst chart shows the relative abundance of the classification results within each taxonomic level. This is very interactive chart. So if you click on each section, for example, if I click on this genus Escherichia, it will show the detail of only this genus and this species. So you can move it around and then click on the interest genus or species that you are looking into. So there are many different ways of presenting data, but this is one of our examples to present your classification output. So this column chart shows the relative abundance of the top 20 classification results within each taxonomic level. So looking at this chart, we can see what is the dominant taxonomy in this sample. You can mouse over to see its description and abundance. And here's the example of one of the dominant taxonomy in this sample. This app generates not only the output report, but also the output files that you can use to create your own graphs or custom analysis. So let's take a look at the output files here. And I'm going to explain the summary CSV file and the classification.txt.gz file. So first, the summary files. This file shows the summarized counts of how reads were classified in the sample. So each unique classification is a row, and then the number of hits and also percentage of total hits are recorded for each row. This file is the same CSV classification summary that you can find under the sample report tab. The next output file is the classification.txt.gz. That name will be slightly different, depends on whether you are using MySick Portal or Metagenomics Metagenomics App, but the information will be same. This file will provide the raw classifier output. So the entries in the file contain the read identifier, which is from the input FASTA file, and then the next row will be the classification assigned to the read with the confidence value, and that will be between from 0 to 1 for each taxonomic level of the classification. Finally, the output includes a PDF report that contains, sample information, classification statistic, and also there is the classification results table that will show you the highest 8 taxonomic classification at each level. There is a pie chart generated from this classification table that show the classification for each level, and it will show any level with more than 3.5% abundance, and the classification with less than 3.5 will be called as others in the pie chart. Let's talk a little bit about the Stickiness Metagenomics Database Creator. This is one of our BaseSpace Lab apps. This app is used to create the custom database, and then you can use that custom database to for the Stickiness Metagenomics app. Right now, the database creator supports FASTA file and TSV input files. The output file generated from this app can be used as a custom database in Stickiness Metagenomics App. And once you click on Custom here, you will have the option to select the custom database that was created by the database creator app. Okay, so for the targeted sequencing, I want to mention in this slide the option for ITS sequencing for fungal classification. ITS refers to internal transcribed spacer regions between ribosome RNA genes. So there are 2 ITS regions, ITS1 and ITS2, and both of which can be used for identification of fungal species. Illumina protocol amplifies the ITS1 regions. This workflow is pretty much similar to our Stickiness Illumina workflow, and primary difference is the amplification of the target locus. If you're interested in, I provide here the link to the tech note that describes detailed information about workflow, library prep, sequencing, and the results using ITS Metagenomics App to perform taxonomic classification using the Unite Fungal database. Another option that I want to include here for data analysis that you can also use the Stickiness Metagenomics app on BaseSpace. And as I mentioned before, you have the option to select the fungal ITS database for classification. Okay, so that was for Stickiness Metagenomics app on BaseSpace and how we analyze Stickiness ribosome sequencing. Now, let's talk about the second approach in metagenomic study, which is shotgun metagenomics. Shotgun metagenomic sequencing provides a way for researchers to survey all genes in all organisms present in the given complex sample. This method goes beyond bacteria and archaea, which is a limitation of the Stickiness ribosome RNA sequencing, and it reveals taxonomic and functional profiles of the microbial communities. So compared to the Stickiness ribosome RNA, due to the PCR bias and also, inability to quantify absolute abundance of the stickiness ribosome RNA, research on the complex microbial community is moving from stickiness ribosome sequencing to more comprehensive functional via shotgun metagenomic sequencing. Some application of the shotgun metagenomics, including agricultural microbiome study, environmental studies, human microbiome studies, pathogen surveillance and identification, and also antimicrobial resistance. Now, speaking of the option to analyze shotgun metagenomics on BaseSpace Sequence Hub, one of the options is our DRAGEN metagenomics pipeline. This will be the analysis option that we are going to talk about in this section. If you are not familiar with DRAGEN, this is our platform, or it's called Dynamic Read Analysis for Genomics. This platform provides accurate and ultra-rapid for secondary analysis of the NGS data. The DRAGEN metagenomics pipeline is available in the cloud through BaseSpace Sequence Hub at this moment. So this pipeline performs classification of the reads using Kraken2 algorithm, and also it is able to detect and quantify SARS-CoV-2 sequences, but also for other common viral and microbial pathogens, and we are going to talk in detail about this app later. Other options for metagenomics on BaseSpace, including Kraken2 Metagenomics Pipeline, MetaFlan, and Cosmos ID. Now we are going to talk about DRAGEN Metagenomics App on BaseSpace. So the DRAGEN Metagenomics App available last year, and this is one of our SARS-CoV-2 toolkits to detect SARS-CoV-2 and perform taxonomic classifications. Now let's go over the workflow of this app. The advantage of this app is using our DRAGEN mapper and aligner, which provides accurate mapping and dehosting option. So with the hosting option, you can remove the human reference genome from the samples. After removing the host genomes, the next step will be taxonomic classification using Kraken2. This app is using the new Kraken2 database in 2020. After the classification, the output will include clonal plots, QC metrics, organism detection report. And one more note here that this app can be used for shotgun sequencing, enrichment panel, and also other targeted sequencing data. So I want to talk briefly about Kraken2 algorithm. So as you know, Kraken2 assigns the taxonomic labels to the short DNA sequences, and the method here is using alignment of the k-mers to the database. Kraken2 is using NCBI RAPSIC database, represented by a series of the clade-specific k-mers, which your reads of interest are queried against. So the way it works is that the k-mer within a read is queried against the database with the k-mer, and that database maps the k-mer to the lowest common ancestor, or LCA, of all genomes. The sequences that have no k-mers in the database are left unclassified by Kraken. The taxa associated with the sequence k-mer as well as the taxa ancestor form a subtree of the taxonomic tree, which is used for classification. So in the classification tree, each node has a weight equal to the number of k-mers associated, and each root to leaf, or RTL, path in the taxonomic tree is scored by adding all the weights in the path. And the maximal root to leaf path in the taxonomic tree is called classification path, which is the one that is highlighted in yellow here. And the leaf of the classification path, which is the orange here, is used for the classification of the query sequence. So in summary, Kraken2 chops the genomes into the k-mers and then link that k-mer to a taxonomic ID. And then Kraken2 also chops the read into the k-mer and search exact hit in the database. it will search for the highest path and then assign that taxonomic ID of the lowest node to the read. So that's how the Kraken2 classifies the read. Okay. Now, for the input of the DRAGEN metagenomics pipeline, there are a few nodes here. The first one is the reference database. So you can see that you have the option to select the mini Kraken2, or you can select extended Kraken2. There's also option for you to use a custom database, and this must be compatible with Kraken2 compatible database. The host reference genome currently only support the human genome. So the next configuration is organism detection report. So in this option that you can provide the list of the organism identifiers via input form. So those pathogens will be included in the detection report. Lastly, in the input form, there's an option for you to change the detection threshold. So if you're interested in detecting SARS-CoV-2, I would recommend keeping the default detection threshold because this is tested internally for SARS-CoV-2 detection. So once the analysis is completed, now let's take a look at the output. So this screenshot shows the output under the report tab in the DRAGEN metagenomics analysis output. This is a summary report for all of the samples in the analysis. The main output I want to point out here is the aggregate organism detection report, which is showing whether SARS-CoV-2 or any organism or pathogen that you specify in the input and see whether they are detected or not. So in this case, you can see SARS-CoV-2 is detected in all 3 samples. The output files used to generate this report will be discussed in the next few slides. So depends on the list that you provided in the input form for organism identifier, those will be included here in this report. So this app also generates the corona plot from Kraken2 output with the percentage of the viral detection. So for example, in this sample, you can see there are 3% of SARS-CoV-2 in this sample. On top of the corona plot, you can see the sample information, which include the CSV version of organism detection report and also Kraken2 classification report. If you're interested in looking at the output of the dehosting option, you can turn off the dehosting option. This is the output when we have the dehosting option turned off. As you can see here, there are a lot of human reads in this sample, which is accounted for like 83% of the total reads. However, if you turn on the dehosting option, with the dehosting on, you can see more clearly that the percentage of the SARS-CoV-2 and other organisms detected in this sample. So in this case, the sample includes 4% of SARS-CoV-2. The next output file is a mapping matrix. So this is one of the advantages of this app is using our DRAGEN mapper and aligner with high speed and accuracy. So DRAGEN performs the alignment to remove the host genomes and then report the mapping output, which is showing here. From this output, you will have better idea of how the alignment works. For example, total map reads, percentage of the map read, or mapping quality. And another output of the DRAGEN mapper and aligner is the coverage. So if you want to check the coverage information, align base and align reads in the genome. Now, let's take a look at the classification output files. So this app generated 4 major output files. Microbe classification report, classification.tsv, classification metrics, and also organism detection report. The first output that I want to talk about is the microbe classification report.tsv. So if you're familiar with Kraken2, this is similar to sample report in Kraken2 output. So this file includes, the percentage of the reads, covered by the clade rooted at this taxon, and the number of breeds covered by the clade rooted at this taxon, the number of breeds assigned directly to that taxon, the rank code, so that indicate whether unclassified or different taxonomic level, NCBI taxonomic ID, and finally, the last column will be indented scientific name. The second output is the microbe classification.tsv. So this file will give you a better understanding of the detailed information for each read that was classified by Kraken2. So this is similar to the standard Kraken output format, which is the tab delimited with one line per taxon. The first column will indicate whether the reads are classified or unclassified. The next will be the sequence ID, taxonomic ID that Kraken2 used to label the sequence. If there is 0, that means the sequence is unclassified. Next will be the length of the sequence in base pair, and the last column will be the space delimited list indicating the lowest common ancestor mapping of each tamer in the sequence. The third output file is the classification matrix, which lists, the classification results, and in which row it will be the number of the fragment count for each taxonomic level. The last output file is the organism detection report.tsv. So this is the output file to check whether the organism, for example, SARS-CoV-2, is detected or not. There is also the number of breeds that was classified to that organism. And this data is actually used to generate the detection report in the summary report tab when we first looking into the analysis output. Okay, so that was the DRAGEN Metagenomics App. That was one of the examples of the analysis method that used for shotgun metagenomic sequencing. Now, I want to talk about the other two applications that will be interesting to discuss because those are two applications that can be used for pathogen detection and also SARS-CoV-2 lineage analysis. The first application is the DRAGEN RNA pathogen detection. The DRAGEN RNA pathogen detection is one of our SARS-CoV-2 data analysis toolkit that provide end-to-end solution for COVID detection and identification. These pipelines are built on top of our DRAGEN RNA pathogen detection app on BaseSpace Sequence Hub. So that helps detect not only SARS-CoV-2, but also other pathogens in our Illumina respiratory virus oligo panel. The library prep kit used is Illumina RNA prep with enrichment, and you use the respiratory virus oligo panel. That will allow researchers to, confirm the presence of SARS-CoV-2 and advance variant analysis studies. Let's talk about the general workflow for this app. So with this app, the reads or the FASTA file will be aligned to the reference genomes. So there are two main references built in this app. The first reference is based on HE38, the human genome, and then Illumina respiratory virus panel. And the second reference genome will be HE38 and 168 viral sequences from Seattle flu study. This is a default setting. So if you have your own panel, you can also select the custom option. The next step will be the human read will be aligned by our DRAGEN mapper and aligner. And then DRAGEN will take the viral read to perform the alignment and variant calling to the virus genomes. After the alignment is complete, then you're going to remove, duplicates or low quality reads, and then it will generate the coverage report. And these coverage plots are then created to detect SARS-CoV-2 and other viral strains. Additional features that include variant calling and then consensus FASTA file generation. So with that consensus FASTA file, you can perform downstream analysis, or you can submit it to the external database. Here's the input form where you specify the project, biosample, variant color, and reference genomes. So if you have the custom genomes, you can see there's a custom option here. So if you use the custom genomes, you can build your genome using the FASTA file or FASTA file with the DRAGEN reference builder. Then you have the option to generate the consensus FASTA file by selecting this option. And you also have the option to include only past variants detected in the samples. Let's take a look at the summary report of the DRAGEN RNA pathogen detection app. The detection is based on the coverage data, especially it's the percentage of pathogen genome with more than 5x coverage. So here you can see almost 93% to 99% of SARS-CoV-2 genome that has more than 5x coverage in these 3 samples. So they are considered as detected. This is example of the bar graph showing the percentage of the viral genomes with coverage 1x and 5x. So in this example, the SARS-CoV coverage, the orange bar indicates the 5x coverage, and then the blue bar indicates the 1x coverage. So it looks very clearly that SARS-CoV-2 samples are positive in this sample. And also, one important note that I want to note here that this is research use only app, so there's no threshold in this case for the sample to be called as detected or not, because again, this is just the research use only. The key output files of this app include the pathogen coverage report, and also as the output of DRAGEN, you're gonna get the BAM file, the VCF file for the variant calling, and also consensus FASTA file. And I'm gonna talk a little bit about the pathogen coverage report. So the pathogen coverage report is showing the number of coverage in each categories. It includes the total coverage with 1x and 5x coverage for each virus listed here. This is actually the data that was generated in the summary report when you see the percentage of the viral genomes with coverage 1x and 5x. And this coverage is actually the coverage per base, but not per read. As I mentioned earlier, you have the option to generate consensus FASTA file. So this will generate the consensus FASTA file in the output, and then you can use that FASTA file for further analysis. Okay, so that was the DRAGEN RNA pathogen app. Lastly, I want to talk about the DRAGEN COVID Lineage App for surveillance use. So DRAGEN COVID Lineage App is our newest addition to SARS-CoV-2 data analysis toolkit that we have on BaseSpace. This is similar to COVID-sick test developed by Illumina, if you're familiar with DRAGEN COVID-sick test pipeline. However, the COVID-sick app is used for COVID detection use, and this COVID Lineage app is dedicated for surveillance. So this app focuses on reporting the lineage and clade for SARS-CoV-2 using most up-to-date pangolin and NextClade software versions. In addition, it can perform alignment of SARS-CoV-2 reference genome, report the coverage of targeted regions, and also detecting the pathogen of SARS-CoV-2 is detected using KMO method, and then perform variant calling and also generate the consensus FASTA file. And lastly, this app is only available on BaseSpace right now. This diagram summarizes the COVID lineage workflow, which is similar to our COVID-sick pipeline. So as you can see here, there are 2 main parts. The first part is the virus detection. So the virus detection is using KMO detection. And it will report the viral coverage to see whether SARS-CoV-2 is detected or not. And the second part will be variant detection and generating the consensus FASTA file. The main difference in this app is that the lineage assignment performed by Pangolin and the mutation annotations and clade report is performed by NextClade, and they are all built in this app. So the final output of this app that you can get the lineage and clade information for SARS-CoV-2. Okay, so I'm gonna go through the summary report so that you have a feeling of how it looks like for this app. So this is how it looks like for the summary report in the DRAGEN COVID Lineage App. The key features here include you can download the combined FASTA file. You can actually take it and drop it into NextClade website or Pangolin. However, Pangolin and NextClade are built in this app, so it will check the most updated version of these two software. So in this example, you can see we have 5 different samples, and 2 to 3 of them are the sample prepared with the ARTIC protocol, and there is a sample from the enrichment and also whole genome samples. So the lineage are calculated and reported out here in the lineage column. The other column is the percentage of the non-N bases, which indicates how many percentage of the genome that are called confidently. I just want to show you an example of the Pangolin aside the lineage. This is basically what happened in the backend and how it works in Pangolin website as well. So you can use the FASTA file from this COVID Lineage app to upload to Pangolin and generate the lineage output data. You can see here, this is how Pangolin output look like in their website. You can see the lineage in this column, and also there is the option to see regional distribution and phylogeny. I also include here the example of the FASTA file with the UK variant sequences that you can download from here. And also I include the GitHub page for Pangolin with a lot of useful information about lineage dataset. But again, Pangolin tool is built in the COVID Lineage app with the most updated version. The next report is the clade report. So this is the output from NextClade. NextClade finds the clade by identifying signature mutations. So this report includes the sample listed, calculated clade, and also listed mutation in this sample, which include substitution by genomics location, deletion, insertion, and also amino acid substitution. And I want to note here that only the variants with the coverage more than 10x and also allele frequency more than 50% are analyzed by NextClade. So the above information are for surveillance information. So if you are using COVID-6, you should use that for COVID-6 for detection. The Lineage app is the research use only and dedicated for surveillance research. However, this app also apply the same method as the KMER detection, and then it will report out the KMER detection report. However, it will not run through QC or run QC threshold, and also it does not apply internal or positive or negative control in this app compared to the COVID-6. The KMER detection report will show you whether the SARS-CoV-2 is detected or not in all of the samples. Okay, so that's all we have today for the webinar. In summary, today we discuss what metagenomic sequencing is, stickiness ribosome sequencing analysis, and we talk about stickiness metagenomics app on BaseSpace. For shotgun metagenomics app, we use DRAGEN Metagenomics App. This is a species level detection of common pathogens, including SARS-CoV-2. For SARS-CoV-2 data analysis, we talk about 2 apps. The first one is DRAGEN RNA pathogen detection. This one will detect the viral pathogens, including SARS-CoV-2 detection and also variant calling. And the next app is DRAGEN COVID Lineage App. This app will detect SARS-CoV-2, perform variant calling, and surveillance analysis. I included here some additional resources that I think it would be helpful. The first one is the metagenomics resources. So I have all of the overview of the metagenomics, introduction to shotgun metagenomics. There is a Kraken2 GitHub page and also some tech notes here. And I include the Metagenomics Part 1 webinar, introduction to library prep and sequencing. And I also have the SARS-CoV-2 resources. There is a very interesting paper in Natural Microbiology that discusses the dynamic lineage nomenclature system for SARS-CoV-2 that I think it will be very helpful. Okay, so that's all I have for today. Thank you so much for taking your time to join us today. I hope this session will be helpful for you.